So our next speaker is actually not Yara Hussein, who unfortunately couldn't join us for this session. Professor Holmes is the head of biostatistics and bioinformatics unit in the MRC Centre of Neuropsychiatric Genetics and Genomics, based in Cardiff, and he has kindly offered to present in this slot very last minute and will present live. So I'll let him take it away.
Okay, thank you very much. Can everyone see my slides? Oh, I assume that's a yes.
Yes.
Okay, because I can never tell whether it's working or not. Okay, so the work I'm presenting here was done by Oliver Didcott, who is a MSc student on our MSc in bioinformatics. He did this as his sort of data analysis project. Okay, so the motivation, as we know, HD age of onset is heavily influenced by CAG length. Increasing CAG length corresponds to decreased age of onset, and it accounts for at least 50% of the variability. So this means that if you're trying to find other risk factors modifying age of onset, so for example, genetic risk factors like the GWAS, the power to find that is going to be increased by taking the effects of CAG length into account. Therefore, it's important to model the effect of CAG length on age of onset as accurately as possible and as wide a range of CAG length as possible. So what we did was we took the Enroll PDS5 data set and used that to compare the age of onset prediction of the commonly used Langbane model to that of a model proposed by Kaplan et al. Okay, so the Langbane model, which was proposed in 2004 by Doug Langbane and colleagues, basically assumes a linear relationship between CAG length and the natural log of age of onset. This fits well for CAG in the range 40 to 55, but fits rather less well for CAG outside this range. So typically the GWASs that I've been working on restrict our analyses to 40 to 55. And of course, it doesn't suggest a mechanism by which CAG influences age of onset. So the other model we're looking at is the Kaplan model. And I apologize for this rather busy slide. I took the figure from the Kaplan paper, which is in PLOS Comp Biol. So the idea here is that the patient inherits a repeat that exceeds a disease-specific threshold, here set to be 38. At birth, the repeat lengths in each disease-relevant cell are sort of clustered around the inherited value. And as you go through the life course, these repeats expand, and they expand a bit more until eventually some of them cross a pathogenic threshold, which is here set at 150 repeats. And then when a sufficient proportion of them cross that threshold, here set to be 20%, disease onset occurs. And as more and more cells, CAG lengths cross the threshold, disease progresses till eventually there's death. And so the Kaplan model assumes that expansion is linear in the number of repeats. So each repeat increases the expansion rate by a certain amount. This gives you formulae for the mean and standard deviation of the distribution of allele lengths at any given time. So, for those of us like me who like mathematical formulae, here are some. They're the only ones you're going to get. I know you're out there. And this model predicts an exponentially decreasing onset curve, which is kind of similar to what you actually see when you look at trinucleotide diseases. So it's a priori plausible. Okay, so what did we do? We took the almost 11,000 individuals with SX rate or estimate of onset from PDS5. We also looked at CC motor age as well, and the results are very similar, so I'm not going to show them. CAG range was 36 to 70. We fitted both models to all CAG, 36 to 39, 40 to 55, 56 plus. Our measure of age of onset prediction accuracy, this quantity R squared, basically it compares how close your predicted ages of onset are to the real ones to what would happen if you didn't try and bother to estimate CAG effect at all. In other words, you assumed that everyone's predicted onset was just the mean of the sample, okay? So R squared of 1 is perfect prediction. R squared of 0 basically means you're gaining nothing. Negative R squared basically means you're actually doing worse than if you just gave everyone the mean, okay? So the Kaplan model has a couple of parameters, the pathogenic threshold T and the CAG expansion rate R, and we just did a grid search of values across these two parameters to find the one that maximized R squared. We chose the repeat threshold for disease to be 36. We also, for comparison purposes, used 37, which was used by the original Kaplan paper on a much smaller sample, which actually didn't have any individuals with 36 repeats. Okay, so this table here. So this column here gives the sample which we used to fit the model. So we fitted a model using every individual. We also fitted models separately for 40 to 55 and 56 plus. This is the estimated pathogenic threshold in repeats and its confidence interval. This is the expansion rate per repeat per year. And this is these are the samples that we use to predict onset in. So the whole sample, 36 to 39, 40 to 55, and 56 plus. You can basically see that when we were predicting onset in the range 40 to 55, you can see that the Kaplan and Langbane models gave similarly high R squared values, and this is by far the majority of the sample. In the range 36 to 39, both models actually gave negative R squared, so basically you're better off not doing anything at all there. In the 56 plus, the Langbane model gave negative R squared. The Kaplan model did quite a bit better, although not quite as well as in the 40 to 55 range. So let's show this graphically. So here's the CAG length along the bottom age of onset on up the side. Each dot corresponds to an individual, and the lines correspond to the various predictors. So the green line is the Langbane model. The dark blue line is just the mean age of onset for a particular CAG length. The sort of yellow line is the new Kaplan model fitted on order data. The cyan light blue line is the Kaplan model fitted just to the range, the CAG length range that we're testing in. So here 40 to 55. And the red line is the original Kaplan model. You can basically see that actually all of them are pretty much as good as each other. Maybe the original Kaplan model does slightly worse. So in the range 40 to 55, you're actually able to get pretty good onset prediction using either of the models. 56 plus, you can see here that because compared to the actual mean age of onset, this blue line here, the Langbane model, rather, the green line, vastly overestimates age of onset, which is why it does so badly. The two Kaplan models, the light blue and the yellow line, do actually keep pretty close to the mean age of onset. The original Kaplan model, the red, does slightly worse, but fitted on a much smaller sample. Okay, so Kaplan's doing a bit better. And for 36 to 39, you can see that all of the models grossly overestimate the age of onset compared to what you actually observe, this blue line here. And the only Kaplan model here is the one that was fitted in the whole sample. We were unable to fit a model just based on the 36 to 39 individuals. It wouldn't converge. So basically here, it's really difficult to predict age of onset via a model. Okay, so the conclusions. Kaplan and Langbane models are similarly good at predicting age of onset for CAG of 40 to 55. And the Kaplan model might be slightly more accurate for CAG lengths bigger than 50. For CAG bigger than 56, the Kaplan model does better than the Langbane model, although neither is quite as good as 40 to 55. When you're in the CAG range 36 to 39, neither model could predict age of onset at all accurately, and both models overestimated it. This is probably due to ascertainment effects. So the conclusion here is that using the Kaplan model to predict age of onset might just enable the inclusion of people with CAG bigger than 56 in our GWAS. Okay, so some further work. Obviously, I'd like to test the Kaplan model in a larger sample of HD individuals with CAG bigger than 55, just to see if it still works. We're looking to apply this model, the Kaplan model, to other repeat disorders. So the original Kaplan paper did do this, but with really small samples. So we're hoping to get some bigger samples. Something I'm kind of interested in is to allow the expansion rate to differ between individuals. So, for example, by including the relevant genetic modifiers in the GWAS, such as the things that Vanessa was talking about earlier on. And I realize I've had to run through this very quickly. If you want to go through these at a slower pace, these results are also presented in poster C6. Okay. Thank you very much for listening.
Thank you very much, Peter, for your very nice presentation. So while we are waiting for the audience, in the meanwhile, I would like to motivate the audience to ask questions. Now this is a next step is to work for the other genetic modifiers. What about lifestyle modifiers? We know that we are working in the European grant.
Oh yes, of course. Actually, if we have those, when I said, I mean, I basically work in genetics, so I obviously think every modifier is a genetic modifier. But you're quite right. I mean, we could, in theory, sort of fit lifestyle modifiers into this model, to allow those expansion rates to depend on those as well. I wouldn't necessarily know which ones to fit in, but people like yourself could help me with that, I think. So that's a really good idea.
We also studied the registry data about the people with homozygosis, and we were surprised because the age at onset of people with two genes affected was similar with people with heterozygotes.
Yeah.
So what do you think could be why there is not a more genetic impact with there is any explanation for? I know it's outside the topic of your talk,
I don't know.
But it's just a curiosity.
I mean, as I recall, the number of people with 2 expanded repeats is quite small, actually, so it's a bit difficult to really infer anything from them. I mean, people sort of treat HD as a sort of dominant disorder, don't they? So having 2 copies of 2 expanded repeats doesn't really matter. So, I mean, in this model, you would basically choose the larger of the 2 to put in. But in fact, whatever model you're fitting, actually, you'd probably choose the larger of your 2 expanded repeats.
I see we have 3 questions now, Esther.
Oh, sorry, I don't. Let me see.
I can read them.
Sorry.
Davina said, did you look at CAG repeat interruptions in your model? The answer is no, because we don't have CAG repeat interruptions for the 11,000 people in PDS5. If someone would like to call them, that would be fantastic, but it might take a bit of time. Leslie says, can you reverse this and estimate age of onset or somatic expansion from the model? Well, that's the idea, kind of. That's what you're trying to do in these models. You're estimating age of onset, and as a byproduct, the Kaplan model, that R parameter is the estimate of a somatic expansion rate per CAG. Of course, how accurate that is would you'd need to sort of do a sort of biological experiment, I guess, to do that. So, Meta says, how did you determine the cutoff on 90 repeats? I'm not quite sure I understand that. So, so we looked at individuals with 36 to 70 because, of course, in PDS5, if you've got more than 70 repeats, you're just coded as more than 70. So we don't know the exact, because of identifiability issues. So that's why we cut off at 70. So does that answer your question, Meta? Sorry, I didn't quite understand that.
If not, I think we'd have to follow it up on the platform. So if you could keep a lookout for that, Peter.